The overall goal of this procedure is to isolate and culture pulmonary endothelial cells from neonatal mice. This is accomplished by first creating a homogeneous cell suspension from freshly harvested lungs. Finely mince the tissue and subject it to enzymatic digestion. Then, enrich for endothelial cells using anti-PCAM1 antibody-conjugated magnetic beads, and seed to culture. Purify the endothelial cell population further by using anti-ICAM2 antibody-conjugated magnetic beads, and amplify these cells in a gelatin-coated flask. The resulting primary pulmonary endothelial cells are mostly of microvascular origin and express specific endothelial cell markers such as VE-cadherin and VEGFR2.
The main advantage of this technique over existing methods, such as siRNA gene silencing, is that it allows utilization of transgenic mice to obtain genetically modified endothelial cells. Furthermore, endothelial cells obtained using this technique are mostly of microvascular origin and as such may be more relevant to some areas of vascular research.
Endothelial cells obtained with this method can help answer key questions in the field of vascular biology, such as those regarding molecular mechanisms of angiogenesis and vascular permeability.
Visual demonstration of this technique is critical as the tissue harvest, digestion, and bead-bound cell washing steps are difficult to learn. Improper performance of this technique can lead to non-endothelial cell contamination and poor cell yield and growth.
For this Dynabeads protocol, prepare 50 milliliters of 0.1% BSA in PBS and mix by vortexing until the BSA dissolves. Then sterilize the solution by passing it through a 0.22 micron syringe filter. Under sterile conditions in the tissue culture hood, resuspend sheep anti-rat IgG Dynabeads and transfer 200 microliters into a 1.5 milliliter Eppendorf tube. In order to wash the beads, mount the tube on the magnetic particle concentrator, or MPC, and allow 1 minute for the beads to sediment. Aspirate the supernatant, remove the tube from the MPC, and resuspend the beads in 1 milliliter of sterile 0.1% BSA PBS. Repeat this wash 3 more times. After the 4th wash, remove the tube from the MPC and resuspend the beads in 500 microliters of 0.1% BSA PBS. Add 10 microliters of rat anti-mouse PCAM1 antibody to the tube and tumble for 2 hours at room temperature. As shown earlier, wash the beads 4 times with sterile 0.1% BSA PBS. Remove the tube from the MPC and resuspend the beads in 200 microliters of 0.1% BSA PBS. Store this preparation of anti-PCAM1 antibody-conjugated Dynabeads at 4 degrees Celsius. To obtain primary cells, anesthetize 3 6 to 8 day old pups. Ensure effectiveness of the anesthesia by pinching the tails with forceps. Dissect animals sequentially. First, pin the pup to a board, and remove the skin from the chest area with dissection scissors. Excise the rib cage to expose the lungs. Now, aseptically excise individual lung lobes, being careful not to dissect the bronchi or any visible connective tissue around the lungs. Combine all lungs in ice-cold DMEM. Finally, euthanize the pups. Now move to the tissue culture hood, remove the lung lobes from DMEM, and place them in a 100 mm tissue culture dish. Aspirate excess DMEM. With sterile scissors, mince the tissue finely by cutting about 100 times. For enzymatic digestion, transfer the tissue preparation into 15 milliliters of warm collagenase disc-based solution and incubate for 45 minutes at 37 degrees Celsius on a rotator. With a 20 milliliter syringe connected to a 14 gauge cannula, triturate clumps of the digested tissue into a single cell suspension. Now, pass the cell suspension through a 7D micrometer cell strainer and wash the strainer with 15 milliliters of IM to stop the digestion reaction. To harvest the pulmonary cells, centrifuge at 400 Gs for 5 minutes and aspirate off the supernatant. Then resuspend the cell pellet in 3 milliliters of 0.1% BSA PBS and transfer to a 5 milliliter round-bottom polystyrene tube. To isolate the endothelial cells, add 22.5 microliters of anti-PCAM1 antibody-conjugated dynabeads and tumble at room temperature for 12 minutes. In the meantime, coat a T75 flask with 2 milliliters of 2% gelatin, aspirate the excess gelatin solution, and allow to air dry inside the tissue culture hood for about half an hour. Once the bead incubation is complete, split the cell suspension equally into 3 Eppendorf tubes and mount on the MPC. Using the MPC system, wash the dynabeads 5 times with about 1 milliliter of 0.1% BSA PBS. After the final wash, resuspend the beads in 1 milliliter of Vasculife per tube. Mix well. Combining all tubes, plate the dynabead cell preparation on the pre-coated T75 flask and bring the total volume in the flask to 10 milliliters with Vasculife. Now, place the flasks in a 37 degrees Celsius 5% carbon dioxide incubator. Change media the following day, allow 1 full day of growth, and then change half of the media every other day until the cells are greater than 70 to 80% confluent. To further purify the primary cell culture, prepare antibody-conjugated dynabeads as shown earlier, but this time using CD102, an anti-mouse ICAM2 antibody. Also coat a T75 tissue culture flask with 2% gelatin as shown earlier. Now remove the T75 flask with cells from the incubator. Aspirate the media and wash the primary cells with 8 milliliters of PBS. Aspirate the PBS, add 2 milliliters of 0.05% trypsin EDTA, and allow about 5 minutes for cells to detach. Tap the flasks lightly to aid cell detachment. Add 2 milliliters of IM to inhibit the trypsin, and scrape any remaining adherent cells. Transfer the cell suspension to a 15 milliliter tube. Harvest cells by centrifugation for 5 minutes at 400 Gs. Aspirate off the media and resuspend the cell pellet in 2 milliliters of 0.1% BSA PBS. Transfer to a 5 milliliter polystyrene round bottom tube and add 10 microliters of anti-ICAM2 dynabeads. Tumble for 12 minutes at room temperature. Split the cell suspension into 2 1.5 milliliter Eppendorf tubes and mount on the MPC system. As before, wash the beads with 1 milliliter of 0.1% BSA PBS a total of 5 times. Now, resuspend each tube in 1 milliliter of Vasculife and perform a cell count. Finally, plate the cells at 800 to 1000 K cells per T75 flask in 10 milliliters of Vasculife. Place the flasks in a cell culture incubator. Change the media every other day until the cultures reach about 80% confluency. The purified mouse lung endothelial cells are ready to be used in experiments. Typically, a cell preparation from 3 6 to 8 day old pups yields 1.2 to 1.5 times 10 to the 6th pulmonary endothelial cells after a week in culture. As expected, these primary endothelial cells display cobblestone morphology under light microscopy and also show VE cadherin staining at cell-to-cell junctions. Using this protocol, the majority of mouse lung endothelial cells express VE cadherin and VEGFR2 as demonstrated by FACS analysis. The red line indicates isotype-specific control, and the green line indicates anti-VE cadherin or anti-VEGFR2 specific IgG.
When attempting this procedure, it's important to harvest only the target tissue, avoid over-digesting the cells, and wash the bead-bound cells thoroughly.
Endothelial cells obtained using this procedure can be used for functional assays in the field of vascular biology to analyze responses relevant for angiogenesis and vascular permeability.
After watching this video, you should have a good understanding how to isolate primary mouse endothelial cells using anti-PCAM1 antibody and anti-ICAM2 antibody-bound dynabeads, and how to obtain pure endothelial cell cultures.